This is like pick a pea. It's like pick a door, but pick a pea. Is this oh my god. Is this actually pick a pea? A troll level made by a wonderful streamer that you should all check out named Mortius. It's called Nice, version 19. If you go off screen, watch your coins. If you get soft locked, blame Box. So this was a contest from another great member of the troll community, Box SMM. Will I get soft lock? All right, let's do it. Wait, what are these? Look at all these shafts. There's some, this level already has ominous shaft. Okay. Oh, there, look at that CP, by the way. What the heck is that CP right there? Oh, stuff's oh, no! I got coins. Oh, I'm up here. Look at me. Hi. No, what does it say? Nice. Not soft locked. Not soft locked. Oh, did I get 69 coins? Oh, that's why the level's called Nice. Oh my gosh. All right. I like it. I'm in. It looks like I can hit this from here. Wait, why would I ever want to go in that box? I guess that's a box that I'm going to end up in, even though I don't want to be in it. All right, how am I going to end up in this box? Wait, how come? Oh, right here is where it will go up. It will go up right here, and then it goes up there. And then, I don't know what's going to happen. I was not at all what I expected to happen. I like how it just comes back with a cannon. Where did this cannon come from? All right, get me out of here. Get me out of here. All right, this is fine. God, that's so stupid. All right, there's like a, can you guys see this? Yeah, there's like a coin down here. I mean, I kind of want to just YOLO the coin. I mean, if you listen really carefully,
my body's ready.
you can hear DGR screams from the bottom of the pit. We gotta go save DGR! Oh, let's go. Let's go. I think that was good. That looked good to me. Let's hit this. I don't know what it's gonna do. Is that bad? That might not even be bad because I'm big. No, 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 no. Look, I'm still fine. I'm fine. Let's go. Let's hit. This is like pick a pea. It's like pick a door, but pick a pea. I'm gonna hit this one. Is this oh my God. Is this actually pick a pea? I'm so happy. I think that this is a pick a door version of with peace. Oh, I want that shiny. Wait. You monster. No. Monster. Oh, I can't get it. No, I need to not do this, right? Oh, and it makes it impossible to go back down there. I see. It makes it impossible to go back down there. Okay. That troll feels slightly unnecessary. Oh my, can I get a sick jump? Yeah, did you see that jump? That was sick. That was a sick jump. Okay, so I do want this, and then we go up here, and then we go over here, and I don't need to do anything with that. We just go here. And then one of these is not going to be a fish, hopefully. Oh, there it is. Nice. Oh, of course, it's the freaking first one. Is there a fish? Oh, I can't jump anymore. Oh. Okay, that's nice. What's up there? Why? I don't understand why there's this, like, one-way gate right there. Wait. Wait. Oh. Do I want that checkpoint? Is this, oh, is this a twice now? Oh my God, this is a twice now, isn't it? This is totally gonna be a twice now. Okay, I'm just gonna stay here then. Yes! No! No! Oh, that's a cool idea though, to have a, like, I guess there, why is it when you load in from the checkpoint, a switch gets hit? Yeah, but when you, Oh, that's why, because you can't get over here. So it's really just probably a thwomp that the thwomp would never get activated because you can't go over that far to the right. This is... God damn it, Mortius. Oh, I'm sorry, guys. I forgot that you can't see what happens down here. My bad about that. I forgot you guys can never see what happens there. I'll just like... Just to show you what's... Oh, don't do that. Oh, wait, am I supposed to go here? No. Now it's like a... Is this an ante? Wait, this is an ante? Oh my god, this is an ante. Wait, no, this is... What is... Okay. Mortius, was that the ante that could be bad? Because I hope there's a worse one than that. Just to show you guys what has been happening all these times. Sorry about that. So basically, this just pushes me off the screen. And then I magically get coins and end up up here. Okay, I do want this, but then I want to go over here again. And then we want to go right... I mean, I think my troll intuition says that I am still going to need... My troll intuition says do the same thing again. Where am I? Wait, I'm getting 69 coins again and I'm back in the ante. What the heck? What? I'm back in the ante. Oh, jeez. Was I getting lucky before? Oh, no. Was I getting lucky before? Because you only have one chance to make this jump. Oh, I think I was getting lucky. I think I, by pure luck, timed this the other times. Yeah, I did this by pure luck. Because you have to get a running start. Oh my god. Okay, I want to do it like that. I got this by pure luck. Oh, come on, let me in there. This is a good top contender. Oh my god. This is a top contender for... How do I want to do this? Like that. How did I do this? Okay, I'm practicing. Okay, that's what I want. Oh! Oh my god. Oh, there it is. I don't know if Carl realized yet, but no matter what he does before he goes into that box, he'll always be at the correct coin count. Oh, wow. So the off-screen shenanigans to sure that are excessive. Wait, so you can collect other coins? Let me see. Oh, I need that. No! Oh my god. Okay. Hoi! Oh yeah, look, there's a 50 down here. Wait, yeah, so check this out. I get a 50 here. Oh, and it like resets my coins so that when I go back into the ante... Whoa, it goes back, so it goes back to 69 still. No matter what coin count you have. Also, where's my music? I believe I was, there it is. Why didn't I hit that block last time? Oh my god. All right, let's see if we can do this ante without ducking, because apparently some people... Oh, okay, you can still do it without ducking. Psh, easy ante. Make it harder next time. Well, that did not happen last time. That did not happen last time. Easy ante. I love this part. Yeah. I haven't gotten a soft lock yet though. Oh, get me out of here. Okay, I mean like this can't be different, right? Like this block can't be different. But I need to get a mushroom somehow. I don't understand how that was different, but I believed in troll magic. I don't want to hit that on and off. I definitely don't want to hit that on and off. Let's see, last time it was, oh god, last time it was the first one, right? It's not going to be it this time, but how can it change? Unless this is this actually just a different area? Is it a completely, wait, why did the switch just change? Why did the switch just change? Why did the switch change? Do I want, I'm going to try hitting this one. Fuck! All right, let's just hit this first one again. Okay. This looks fine. But, oh, okay. Bum, 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 bum. I think we're going in the key door. Is this the end? Nice. No, this is gonna be off screen. Why is this off screen? No, you don't give me 69 coins. Don't give me 69 coins. Oh, okay. Oh, that was cute. That was cute. That was a really cute level. I thought I was gonna go back into that ante somehow. I love that level, Mortius. That was really fun. Oh, you have it behind the scenes? Oh, you have it behind the scenes. Sure. Oh, you have it in, oh my God. Look at the, this is really cool, Mortius. Look at the contraptions that Mortius had to make. These are like, you can see all the coins and everything. Wait, so Mortius, was it a twice and not two different areas? I don't understand where the launchers came from, right? I mean, I guess these are the launchers. I don't understand why they like fell here, or did they fall here? What the heck? At least 60% of the off-screen contraptions are just to make the coin count work. Look at all these coins. So I want to know, how do you have, is it the case that I could not have hit the Kaizo block on the first pass?
Correct. So if you look, that spike ball actually activates. So look sort of down to the right of that. You see that note block?
Oh, down here, yeah.
So that note block shoots up and hits the spike block to activate the Kaizo, and then that bomb explodes it.
I see. So the spike ball drifts to the right, the note block comes up from below it and hits it up into the hidden block?
Correct. You can actually,
that's so cool.
On the second time through, you can actually see it. I don't know, since it's so short, I don't know if you want to play it again to see that, or if you don't have the time, but you actually can see that contraption occur in the second time through.
Okay.
And also, you were asking about the clop at the top left.
Yeah.
Again, if you wanted to replay it real quick, that's actually to give you a hint if you miss the pit mushroom and just go above without collecting the mushroom. When the switch state changes, that claw goes over and grabs the mushroom as kind of a cute little hint,
it like grabs it out of the pit? Oh.
It grabs it out of the pit. That's what all of those tracks are for.
Oh
it's
my
actually
God.
really cute if you want to go check it out.
Yeah, actually, I do want to check that out. That sounds awesome. Okay, so I was convinced that it was two separate areas. That's even crazier to me.
It was a requirement for boxes contest that it had to truly be one room.
Wow.
Which is why the off-screen shenanigans are so ridiculous.
They're quite ridiculous. That was really cool. Let me see if there's anything. If there's anything else you want to tell me, let me know. Or else let me see if anything like jumps out at me.
I mean, pretty much, if you see all of those like blue platforms to the left, those are all set to various different timings just because. So I admittedly am not the strongest troll maker from the trolls itself. So my levels focus a lot on theming. Like Pot of Troll, the level I made a couple years ago, had a lot of trolls that were explicitly rainbow themed, which was really hard to wrap my brain around.
Yeah.
Because I want the player to like feel like they're going through an experience, since the trolls themselves aren't anything revolutionary in my levels. You know what I mean?
Yeah, that's such an important part of making a good level, though, is having the theme be good.
Exactly. But so the reason there are so many of those blue platforms is because the timing of the 69 coins is actually different depending on where in the level you get it. Like if it's at the very beginning, it's a very slow, like, oh, 1, 2, like 61, 62, 63, 64.
I see.
But if it's later in the level, it's a much faster track, so it's to get you back into the ante a little bit faster.
I noticed it was fast, yeah, because you go into it so many times in that second path on the right side.
Exactly. So there's actually 3 different timings. It's like the first time you're in it, and then the second time you're in it, but before it's an ante, and then the next time, once it's an ante, I want to get the player into it as soon as possible to reduce frustration. So that's what all of those blue platforms are for there.
Interesting. I think I can see
the,
it sort of just looking at them here.
Yeah.
How like.
And then the top right. Sorry, go ahead.
No, it's good. I was just trying to see like you come out this other one. Like I'm just trying to imagine you land on this other blue platform, and I'm just sort of like imagining it would go down, but then you'd like hit this other one, and I don't know if I guess that one would take you back up and.
Yeah, and there's even the very bottom one as well, is because if you jump in a certain way, like if literally frame perfect, you can end up soft locking yourself down there. So
that's where the soft lock was. Okay.
That second one is no, not anymore.
Oh, okay.
Actually, the reason that second one is there is to catch you. So actually, there is a soft lock possible, but you have to get 2 frame perfect jumps. Because that second platform is there to catch you if you get the frame perfect jump the first time.
Okay,
so there's really... like a frame perfect jump that you have to make on each of those platforms to successfully soft lock yourself.
Nice. Well, I'm sad I didn't do that. Cool.
Everyone in chat's talking about an ice block. I don't even know what they're talking about.
Oh, this one, right?
What?
I have the pointer on it now.
I don't know, honestly. I couldn't tell you.
Maybe it had a purpose. Maybe it just got stuck there.
Yeah, no idea. And then obviously up at the top right, you've got some ridiculous shenanigans just to allow for those launchers to actually drop down at the correct timing and everything like that.
And the launchers would only drop down when the switch state was hit, right? Or like the other extra ones would only drop down.
They only spawn in.
Oh, so there's blocks behind them.
There are blocks behind them.
I see. You just can't see them on this. Okay.
Because you were like, how could these 2 blocks possibly be different? So you can actually see.
Oh, there's 2 actual blocks. Oh,
there
I just saw it. Oh my god, I just saw it.
That bomb right above them.
Yeah.
Is exactly that.
Does this bomb like? Because I see it would just walk, right? Like. How does it end up hitting just the, like, what's the path that this bomb takes when it spawns in?
So there's one bomb with a blue, like red right above it, but then look to the right, there's a bomb with a blue on-off state. So if it spawns in one on-off state, then the bomb to the right walks over and destroys the bottom one.
Gotcha.
But if it's the other switch state, then it destroys the top one because of that higher bomb.
How does the higher bomb hit the top one? I'm just kind of curious.
I would love to tell you I remember.
Oh, yeah, it's been a while. It doesn't. So it looks to me like it will walk off the edge and maybe hit the spring or maybe not, and like just.
I know it has to do with the winged block to the left of it, but I don't remember how exactly. I don't remember where exactly it hits it. I know the wing block does, but I don't remember how.
Maybe the wing block just crushes it against like something or something. Yeah. Okay. Okay. All right. Really cool stuff, Mortius. Thank you. Okay, we're gonna skip that, and now there's apparently a hint over here. There's a hint over here. Oh, there comes the claw. Oh my god, I love this. And the claw fishes out a mushroom. Look at that. That's a little hint. Hey, there's a mushroom down here. That's really cute. I love that. And I guess it does it the next time too, but in the other time it fishes it out. It fishes it out of the other track. That's the cool hint. I like that a lot. Good shit, Mortius. Good shit. And Mortius has also been working on another level, which was described as like forged in the post 5 golden swings rage fires or something like that. That we might maybe have to look forward to in the future. That I have a feeling is going to be significantly worse than this. By the way, I made a new science YouTube video. If anybody is interested, you can check out Laurel's Lab on YouTube. I'm making sort of like more shorter videos because it's something that I can actually do. Like I made this video in a day. This was 2 hours of lab work, but like still 8 hours of video editing. You should check it out on my science YouTube channel. I'm also making little silly things for TikTok. Should that be your thing as well?
Let's grow some bacteria.
Delicious yeast soup. This is what I did in the lab
this is bacterial growth
yesterday.
Media. I'm going to be sterilizing these tubes with this flame and then adding in this bacterial growth media.
So I'm growing some bacteria for my experiments. Next, I'm going to add a little bit of the antibiotic kanamycin because I only want the bacteria that are resistant to kanamycin to grow. These bacteria that I use have a special little thing that makes them resistant to that antibiotic.
Mix it all up. Frozen bacteria.
This thing's cool. It's a negative 80-degree freezer, super cold. And these bacteria will stay alive in it for literally decades.
Frozen bacteria.
Each one of those little
So
bacteria.
after this inoculating loop has been sterilized, die bacteria, die. Then I take a little bit of the bacteria that are frozen in this tube.
That thing was too hot and it
I'm
killed some
gonna
of
put the
them.
bacteria in one of these tubes, but I'm not going to put them in the other one. This will show that the liquid itself isn't contaminated with bacteria.
I literally tripped right here.
So this is gonna shake overnight. It shakes so that the bacteria.
Oh god, I was holding a tripod with the camera on it, and the bottom part of the tripod snagged on the freezer that I was walking by. You really like the style of editing? Yeah, it's just I'm just having fun with it. It's the sort of editing that I can do in like a few hours. Why do you heat up the flask, the top of it, before putting the cap over it? In case any bacteria in the air landed on it, it sterilizes it. Carl dropped the bacteria on his gloves. I do at some point, actually. Yes, it's non-pathogenic E. coli. Why do you only sanitize the top of the test tube? Well, the top is the part where the air can get to it, so that's the part I'm the most interested in. You have a dedicated mixing machine. Yep, it shakes them all night, and it helps them get air mixed in. What's the funniest thing you've left on the mixing machine? I could find the video and show you, but one time I put these things on it, and like I thought they were gonna roll off, and they just rolled almost off over and over and over again, and it was hypnotizing to watch because they never actually rolled off. Should the depositing of the tubes be done in a sealed environment? No. If they were pathogenic, then yes. So like this lab that I work in is called Biosafety Level 1. It's the lowest biosafety level. None of these things can infect people. Like you could practically drink them and still be fine. Obviously you shouldn't do that, but the fact of the matter remains that they don't infect people. I also have another room that I work in that does have like viruses and stuff. Like for instance, we just got a shipment of dengue virus. And so for working with dengue virus, I have to work with it inside of it's called a biological safety cabinet or a hood, and it has like negative air pressure, so that I have to like stick my hands into the hood and do everything like in this sealed off chamber inside of the hood, and there's like an airflow that keeps anything from getting out of it. You also have to like bleach everything all the time or whatever, and that's called Biosafety Level 2. Now there's 2 more levels past that, Biosafety Level 3 and Biosafety Level 4, that are even scarier. Biosafety Level 3 I've never worked in, and you basically have to have a suit on or whatever, because like even if you breathed it in, this is things like tuberculosis or whatever, or COVID is also BSL3, Biosafety Level 3. If the virus were to get out, what's the worst that could happen? Nothing bad with anything I work with, which is the point. And yeah, Biosafety Level 4 is like, it could be a real bad problem. Like Biosafety Level 4 is like, yeah, it's like Ebola. Like if it were to get out, then it could be a serious problem. And so there's only a few biosafety level 4 places that exist. They have an entire facility, and that's the sort of thing where like you go into the decontamination chamber and you get like fully sterilized or whatever, and then you go into the lab, and in order to leave the lab you have to again be decontaminated or whatever. And you need like a full body suit and all sorts of shit, and I've never even seen one of those. Yeah, so anyway, I'm growing these bacteria.
Constantly mix with the air, because they actually need that, like those bubbles and stuff, they need all that air to breathe and grow. Something close to breathing. It's fine. Come back the next day and we'll see what happens. All right, let's see what we got. Yeah, there we go. See how the one on the left that we put bacteria in is now full of bacteria.
I need to play Final Fantasy.
My throat is also full of bacteria.
I have been choking for like the last few days.
All right, next we're gonna get some of these tubes out.
Yeah, it takes a whole day. Yep, 24 hours for them to grow that much. Yep. And so like the cloudiness is trillions of bacteria.
All right, we're gonna put these in the centrifuge that spins them really fast. And this thing is gonna spin them around super fast, so we're gonna come back in 5 minutes.
A science GoPro. It would prevent me from tripping.
This is spinning them at 4,000 times the force of gravity, relative centrifugal force. Please do it.
This is a centrifuge.
Thank you. Start.
Why do spin things? You will see why do spin things.
All right, be back in 5 minutes.
Because I don't want to inject plants with bacterial growth solution.
Let's go. All right, you see how the bacteria are all down in a clump at the bottom there. We're gonna dump everything else out because we don't need it anymore. Get out of here.
Tubes go burr.
We're gonna remove.
It separates them based on density.
There, so we just have that nice bacteria.
So that lets me get all the bacteria.
And then we are gonna mix them up with some infiltration buffer. So this is a buffer that will help the bacteria infect plants. All right.
So this is a special type of bacteria that infects plants.
And then add more of that infiltration buffer.
And inside those bacteria, I've put a gene for one variation of the vaccine that I'm working on. And so the plant leaves will make... I spilled it.
Definitely just spilled that everywhere.
If you're wondering what it looks like when I spaghetti in the lab, that's what it looks like.
Whoopsie! Plants.
Yeah. So I made that growth chamber there to grow these plants because our air conditioner died in our normal growth chamber.
Suck up some of that bacteria. All right, I'm gonna make a tiny little prick on the back of the leaf with a paper clip.
Are you not survival free? Exactly. That's why I'm level 1, and I'll never do anything other than level 1.
Whee! Yeah.
I think it's cool how you can see it going in.
All right, now all the leaves have been injected, so these just need to go grow for a couple of days. All right.
So it takes about 4 days, and then those leaves will be packed full of... So these experiments are all part of a project to make a norovirus vaccine. I'm actually trying all sorts of different vaccine candidates, and this is just one of them. This particular vaccine involves making the protein that allows norovirus to attach to our cells, which is similar in some ways to the COVID spike protein that we all probably know about at this point. So all of these little colored blobs are the protective shell that surrounds norovirus, and most viruses have something like this. So what's interesting is every single one of these is the exact same protein. It's 180 copies of a single identical protein, but if you just throw 180 of these, or enough of these into a solution, they all stick together in this exact same pattern. Well, it's very close to the exact same pattern. And if you look at it, you see here there's 5 of them right here, so it makes this little pentamer thing here or whatever, and then it sort of makes these it looks a lot like an icosahedral 20-sided dice or whatever, but I don't know how many sides this one would be, but inside of this would be the genome of the virus. And so when this little ball goes out into when you eat it, because it is floating around in the air, or when somebody vomits, you'll vomit these out, or if you have diarrhea, from getting infected with this, then you'll poop them out and whatever. And if somebody else comes in contact with one of these, it goes into your stomach, and then this thing is specially made to be able to survive stomach acid. Forbidden bubblegum ball, exactly. Yeah, it's a biological polymer. People use these for as nanotech nanomaterials because the self-assembling nature of them to all stick together in this way, you can actually exploit that. Maybe you could package a drug inside of it, or a medical treatment or other things. God, that's delicious. Tina Hacks asks, how new is targeting specific proteins in vaccine tech? I feel like I hadn't heard of that technique while I was reading white papers about the COVID vaccine. So it's been around, the idea has been around definitely since I think the first one was the hepatitis B vaccine, which was in the late 80s. They produced the hepatitis B surface protein, which is one protein like this, in E. coli. And just that one protein was used for the hepatitis B vaccine. These are called subunit vaccines, and that was the first time that had ever been used. But the first time that a vaccine that makes a virus shell like this, in this sort of way, was ever used was the human papillomavirus vaccine, HPV vaccine, which I think was 2004 or 2006, something about that. And so, but there really aren't very many vaccines that use this technology. And of course, the messenger RNA vaccines that just came out for COVID are like a really specialized form of this, where like your cells are just making like this one protein, and then your cell will make a bunch of them. And then in the case of SARS-CoV-2, the virus that causes COVID, this protein doesn't stick to itself like this and make the shell of the virus. Instead, it sticks into a membrane in your cell. And so like a bunch of the SARS-CoV-2 spike proteins will stick in a membrane, and then it'll like bud off of it. It basically steals part of your membrane, and that is the protective coat for the virus or whatever. But anyway, let me see what I talk about next. However, this vaccine has one key difference. I've gone in and removed parts of the protein that vary the most between different strains of norovirus. Okay, so the key difference between like what is happening for the COVID vaccine is like they just make the protein, right? Like they give you the spike protein and that's the vaccine. In the case of norovirus, if I were to give somebody just the spike protein, which is this thing, it's not really called a spike protein for this, but it's the same idea. It would protect them only against one strain of norovirus, but so the result is that hopefully it will create a vaccine that can simultaneously target many more strains of the virus because they're okay. So you know how like for COVID we have, you know, alpha, beta, delta, omicron or whatever. They're basically all 99% the same. Like there's only tiny differences. Each one of these little fucking things is a different strain of norovirus, and these motherfuckers are like 40% different, right? So they're only like 60% the same. So like within just like one of these, you have like everything we have for COVID, and then like you come over here and it's like it's not even the same thing, and it's not even the same thing, and it's not even the same thing, it's not even the same thing, and all like we get on average 5 different norovirus infections throughout our lives. So the average person gets norovirus 5 times in their life, and it's like you're gonna get one of these, then you're gonna get one of these, then you're gonna get one of these, then you're gonna get one of these. So it has like, to say that it has more variation is like an understatement. Like if SARS-CoV-2 keeps infecting humans for the next 1,000 years and keeps evolving and making new variants, that's like what norovirus has already done, which is why it's so hard to make a vaccine for, because you can make a vaccine for one of these and then you just get hit with another one and the vaccine doesn't do anything. So what I'm trying to do in these vaccines is I've taken this protein that makes this protective shell for norovirus, and what I've done is I've gone in and I've snipped out the pieces of it that differ between all of these strains, because there are some parts of it that they all have in common and some parts of it that they don't have in common. And if you just give somebody like this entire ball for the vaccine, which it makes a great vaccine against that one strain, it's sort of like the parts that your immune system sees the most, like the parts that it makes an immune response against the most, is really the part right at the tip. And that part right at the tip is the most variable. And that makes sense, right? Because like the reason that these other strains evolve is they differ in a way that they can evade your immune response. Because if they didn't, then they wouldn't exist anymore. That's how evolution works. Literally just the tip, yes. And so the reason that these viruses have been able to exist and evolve is they change the part that our immune system sees most commonly, and thus can continue to infect us. Because if they couldn't continue to infect us, then we would all generate herd immunity and they wouldn't exist anymore. So what I'm doing in this vaccine, and I don't have an animation for this, and it would be cool to have an animator draw stuff if anybody's interested, but like the reason I don't have an animator is it takes so long and or whatever, and like I can pay some, but I can't pay that much for all of these videos. But what I've done is, someone says, can you give people more than one complete spike ball? So that's what the HPV vaccine does. So what the HPV vaccine does is it gives that, like Gardasil 9 is 9 of these spike balls for the 9 most prevalent strains. And that is helpful. Norovirus is complicated because there's just so gosh darn many of them, but it might be possible to do something similar to that too. The problem with doing that is that it is very expensive. So the problem with Gardasil 9 is it is the most expensive vaccine on the market. And even if you chose 9 of these, there's still so many more, but there's an even bigger problem. Norovirus evolves so frequently, more than HPV, that even if you vaccinated somebody against like the 9 most prevalent strains, in another few years, it might might be a different virus, and so then you would need a new vaccine again. That still might be good. That still might be better than what we have. But the concept that I'm trying to make is that it is to make a vaccine that won't just protect you against the current common strains, but it will protect you from future strains. Now, it's possible that the virus can evolve around that. It always is possible. But it's like we have plenty of vaccines where viruses don't evolve around them. So, like, for instance, measles or whatever, like the measles vaccine worked. It worked 30 years ago. It still works now. So it's probably possible to like evolutionarily pigeonhole the virus so that our immunity is so good that it can't evolve around it. Like, it doesn't have enough time to evolve around that. Why do some viruses mutate faster than other? It's very complicated, Hippo. Like, the simple explanation is that the way that their genes are copied or replicated is more error prone. So like there's a biological enzyme that comes in and it'll make a lot of different copies of their genome, and a lot of viruses, like, they have their own enzyme that does this. It's part of their genome. So it's like they have part of their genome that makes something that then makes more of their genome. And so because the virus controls that, it can make it better or worse. And so a virus can evolve to be more evolvable. It can evolve to be more mutatable. But if a virus mutates too fast, then it will not be functional. Like, so it won't be able to maintain what works. It'll lose that, and then it'll sort of like evolve itself out of existence. So there's a balance there. Yeah, it fizzles out. So there's a balance there between changing enough to, be able to evade our immune system and the immune system of whatever it's affecting, over the course of decades, but not lose what already works. The plan for this norovirus vaccine, and the same plan me and other researchers are working on for HPV, and me and other researchers are working on for influenza, is to make a vaccine that is broadly protective enough to protect you against current viruses, but also future ones, because then you wouldn't have pandemics in the first place. If people had made a vaccine like this against coronaviruses before COVID ever happened, then maybe the pandemic would have never happened. So anyway, it would make vaccines cheaper to just have like one simple thing that is like the parts of the virus that don't change, because then you don't have to vaccinate someone with 10 different viruses or whatever. So it would be cheaper in that sense. It would also be better if you could make it work, but there are problems. What are your thoughts on Walter Reed possibly developing a pan-coronavirus vaccine? G-Bads, that's just so the way that they did it isn't the same way that I'm doing this. Theirs is just the normal thing, and it just happens to provide some protection. So, it is possible to, like, the strategy that I'm doing is don't even show your immune system the parts of the virus that are highly variable. But another way to go about it is show your immune system everything, but the vaccine itself may be like shaped in a way that one shape is easier for the immune system to see. Something like that. You can also like cover the parts of it that you don't want. Like, for instance, some people will stick antibodies onto one of these things and block the parts that you like don't want your immune system to see. Or maybe you'll mix a chemical in it and the chemical will stick to those parts. All of these are different ways of giving your immune system a single virus protein, but the result of what your body makes an immune response against will be different. So anyway, these are all things that different people are researching. Is there any benefit to humans for viruses to exist? Would it be good if we managed to eradicate them? So Smallbert, like 7% or something like that of our DNA is viruses. It's thought that, like, for instance, the placenta evolved from virus genes. And it's also thought that one of the ways that neurons communicate with each other is actually like our neurons take some of the signals that they want to send to each other, and they put them in a protective shell that, if you look at the gene, it was clearly stolen from one of these virus shells, or they're called capsids. So like, clearly viruses have been beneficial to us, but I don't think we need most of them anymore. There's other research that's like, so there are some viruses that kill bacteria, and those they're called phages, and they live in our intestines, and those phages actually help control the bacteria that live in our guts. And they're probably very important. But no amount of vaccines that we make for human infecting viruses will ever affect those. So like, I wouldn't want to get rid of every virus, but I would happily get rid of every virus that commonly infects humans. I don't think that any virus that commonly infects humans is doing anything useful for us. Though who knows, like if our immune systems have nothing to fight, then there's at least a case to be made that, well, maybe we'd get more allergies or something. But like, would I rather have COVID or allergies? Like, fuck, give me the allergies. And of course, we can deal with that problem later. What about combining your approach with having multiple different balls? One ball, all the similarities of half the strain. Totally exactly what I'm doing, Kimura. I haven't told you this, but I've already done that. So what I did in here is, see how there's Genogroup 1 and Genogroup 2? I've made one ball of similarities for Genogroup 1 and one ball of similarities for Genogroup 2. And in this, literally yesterday, I have now this plant that has those sitting in our growth chamber right now, and in 3 more days I will extract the virus balls from it and do some tests on them to make sure that they've formed properly. But I've already tested them and I'm pretty sure that it does. And so the next step after this is to vaccinate mice with it. It'll probably take 4 months or so for that to happen. And so, science is slow. You need to wait for the mice to make an immune response. Then I will take antibodies from those mice and test to see whether they stick to a whole lot of different types of these different noroviruses. So I made a different video. So this is my other video that shows how I will test these at the end.
Have you ever wondered, how do we design vaccines? Like, how do we know it will work before giving it to billions of people? One important test is this thing, called an ELISA. What an ELISA does is it's used to test whether 2 biological things stick together. In the case of a vaccine, we want to know whether the antibodies in your blood stick to the virus that they're supposed to protect you against.
So this test is like, once I'm done with these new norovirus vaccines, making these different shells and whatever, then I will use this test and a bunch of other ones. This is just one that I've made a quick video on. Yeah, good face to pause on. This is a test that will allow me to see whether or not the antibodies made by a mouse or a human or anything that you vaccinate with will be able to stick to all these different viruses.
And if they do, then that's a good sign that the vaccine is working properly. So in this experiment, we start by putting in a solution that is essentially little pieces of viruses. By the way, the particular virus that I'm testing here is one that you've probably had before when it's coming out both ends. Except in this case, I didn't use real norovirus because we don't want to get sick doing these tests.
By the way, a little side note here. Some people always ask me like why I work on norovirus. Well, it's a virus that has like heavily evolved so that there's like a billion strains. So like in principle, if we can make a vaccine like this against norovirus, you could probably do it for just about anything, though HIV is even worse. But the other reason is I actually got extremely sick with norovirus. I got really sick with norovirus when I was a kid. I literally barfed and crapped everywhere in the middle of school when I was in I think it was when I was in 3rd grade. I was very young, and it traumatized me. So I was afraid to go to the cafeteria and eat because I ate in the cafeteria and then I just threw up everywhere in school. And so it literally, I think it might be where my anxiety disorder started actually, because I became terrified of going to the cafeteria again every day. So basically every day I got severe anxiety because I was afraid of that happening again because it was so traumatic. So I wonder if some of my anxiety disorder was caused by norovirus. So that's one of the reasons why it's fueled by vengeance.
Instead, I used what could be thought of as just the empty shell that surrounds and protects the virus. All viruses have a protective shell around them, and it's only the stuff on the inside,
that's a 3D printed
made out of either
virus
DNA
model
or RNA, that cause you to get sick. We put the solution in these tiny little wells that are made out of a
So I put each of those different strains of norovirus at the bottom of that plate.
Material called polystyrene. Polystyrene is really interesting because it sticks to proteins. And since viruses are mostly made out of protein, the viruses will stick to the bottom. Next, we wash off the remaining bits of the virus and add a solution of milk. This makes it so that nothing else can stick to the plastic. Next, we add serum from blood, because if a vaccine has worked properly, your blood will be full of antibodies that are specifically able to stick to whatever virus you've been vaccinated against. You can also test saliva or mucus this way, because those would also contain antibodies. But if you look at what's going on here, you can't see anything happening. Viruses and antibodies are both too small to see. So how do we know if the antibodies in the serum actually stuck to the virus? First, I washed off the extra serum because we only want the antibodies that were actually able to stick to Norovirus. All the other ones will get washed away.
So somebody says, why does the milk have the just normal ass polystyrene? I think it's treated, Tina. I think the plate is made out of something called polystyrene, and I think it's treated so that when the norovirus shells or whatever, and you could use real norovirus too, or whatever virus you want, any protein, it will it's called electrostatically interact. So basically the charge of the protein is, I guess, opposite the charge of the polystyrene, so it will stick. Yeah, it's an ELISA. So first you stick the protein down, and then you put on the antibodies from, blood, saliva, mucus, whatever. And if there are antibodies in that blood, saliva, or mucus, they will then stick to the virus that's sitting at the bottom of this plate. The reason that you add milk in between is because we don't want the antibodies from your blood to stick to the polystyrene. So the milk is like random decoy proteins that will take up all of the remaining spots on the polystyrene, and then the only way for the antibodies to stick is if they actually stick to the virus. They're not just going to stick randomly, non-specifically to the plastic. And of course, you can do controls where you test to make sure that this is right. Like if you don't add the virus thing, then you won't get any signal and that kind of stuff. So you have all sorts of controls to make sure that all those things work the way I just said that they work.
Then I added a really cool substance that sticks to antibodies and turns blue. So if there are any antibodies stuck to the virus, the well will start turning blue, and the more blue it gets, the more antibodies there were.
So that part's pretty cool. Basically, you've got a virus and then you've got an antibody stuck to it, but you need some way to actually see that or get a readout that you can quantitate to know like how many antibodies were there in your blood. Do you have a strong response or not? And by the way, this is how like they do the responses to figure out like, hey, 2 doses of COVID or 2 doses of the COVID vaccine or 3 doses of the COVID vaccine produce a higher antibody response. It's literally this test, and they'll get a stronger readout at the end of this test if you've had 2 doses or 3 doses or whatever. But the way that you actually detect it is interesting. It's so weird. There are antibodies that will stick to literally freaking everything. And so like, let's say this test is using your serum, and so your serum has human antibodies in it. Well, there is an antibody that sticks to human antibodies. And so it can be like, originally you can make these things with by vaccinating a goat with human antibodies, and then that goat will make its own goat antibodies that stick to human antibodies. And you can do that with everything. But these days now you can just make them synthetically. So like it's known how to make an antibody that will stick to human antibodies. And for instance, I can make it in plants. Like you don't even need a goat. You can just make it in plants now, or you can make it in whatever. But those antibodies have had God, how do I say this simply? It's called horseradish peroxidase because it's an enzyme that was discovered in horseradish that will cause a chemical reaction that causes a color change, and they have stuck that horseradish peroxidase onto an antibody. So if I mix that antibody that sticks to human antibodies with this horseradish peroxidase thing stuck to it, it will make these little wells turn blue if they contain human antibodies or whatever. So it makes it turn blue if the antibodies are there and not if it's not there.
Finally, I added a solution of acid that stops the color change from happening, and then I took it to a machine that reads exactly how much color has been made. In my case, I tested 4 different ideas for vaccines that I've been trying to make against norovirus. 2 of them worked pretty well, and 2 did not work at all.
So this was my first attempt at making this norovirus vaccine thing, and I tried giving the mice, remember that like ball thing or whatever? Like if you think about the part if you think about like the part of that like shell or whatever, if you just think about one part of it, there's like the tip that changes a lot, and then there's sort of like middle segments and lower segments. I tried vaccinating only with the tip, only with like the top segment, only with the like lower segment, only with the base segment. And 2 of those worked kind of well, and 2 of them didn't work at all. It seems like there's some part at the base that works really well and some part in the center, but the other ones didn't work super well. And so the next idea that I'm trying right now is like, okay, I'm gonna do the whole thing and just like take out a few little pieces rather than only giving the mice a few pieces in the first place.
So next, I need to figure out why those 2 didn't work, and I'm going to see if I can come up with any new ideas to try to see if I can make this potential vaccine even better. Though in reality, there are dozens of other tests that also need to be done to figure out whether a vaccine worked well or not, but this is a very important first step.
How do you modify the protein to your liking? I can just delete the sequence that I want. Like, it's all ATCs and Gs or whatever, and those ATCs and Gs will build the protein. I can just send a new sequence to a company with the parts that I want removed, and they'll just send it back to me. But sometimes I don't want to, because that's expensive. Sometimes I don't want to have a company do it for me. I use these little things that can be thought of as little molecular scissors that will cut out a piece of it from the gene itself. So once that piece of the gene is cut out, then I put it back in those bacteria, inject it back into the plants, and then the plants make it with that piece missing, if that makes sense. So there are tiny little, they're called enzymes. They're tiny molecular machines that will recognize a certain part of a DNA molecule and cut it. And then there are other ones that will paste it back together. And so I cut and paste it at different times. Yeah, it's like CRISPR. CRISPR is honestly harder than what I use though. I mostly use restriction enzymes and then a technique called polymerase chain reaction, or PCR. I should do a video. Honestly, I think that's the next video I'll do. It's hard to do that video because it's all invisible. But I'm gonna do how to cut and paste different pieces of DNA in and out using polymerase chain reaction, I think is what I'll do next. That's actually a really cool one. Do you do several rounds of GNA, or do you just insert made to order genes? I do both. If I can do it myself, I do it because it's cheaper and faster rather than waiting for a company to make it. But if it's really complicated, then I'll have a company do it. Do you do it in small amounts and it copies itself? Yes. So you do it once, and then you inject it into some bacteria. Well, zap it into some bacteria, and the bacteria will then replicate it trillions and trillions and trillions of times. Just use MS Paint. Do viruses become more resistant? It just depends. Viruses become more adapted to their environment, whatever that environment is, to the extent that they can. Like a virus can't do anything. Like, people are always like, oh, are these viruses going to become airborne? And they take every property of a virus and they imagine like a super virus that has all of them. But the reality is that like a virus that kills everybody instantly but is also airborne but is also like never degrades, and also like these things are mutually incompatible, thankfully. But also like it makes sense, right? Like if a virus wiped everything out, then it would have no more hosts and it'd be gone. So yeah, this has been around since the 80s, or really the 70s. People have gotten much better at it now though, and like having a company synthesize any gene for you really quickly and like being able to sequence it really fast is definitely like the last 10 years. Peanut butter, norovirus is not very heavily studied at all. I would say there's like less than a dozen labs in the entire world that study norovirus vaccines, and it's probably less than that. There really aren't very many at all, which is one of the reasons I was attracted to it. It's something that basically nobody is working on. I know you're a postdoc, but do you think about becoming a professor? Yep. My PhD program is coming to an end, and I'm kind of paralyzed. I like teaching, though. Yeah, I relate, dabble. I like doing research in the lab, but I also really like teaching, and I really miss teaching. But being a professor is a nightmare. Like, if I were a professor, there's no way I could stream. I just wouldn't have time. I don't want to do all the administrative bullshit that goes into being a professor. I like working with students in the lab. I like doing lab experiments. I like coming up with projects a lot. I really like coming up with different projects. So I don't love... A lot of lab work is very tedious and repetitive, and it takes forever, and you have to do it over and over and over and over and over and over and over again, and then it doesn't work, and then you do it all again. Yeah, Daisy's like, there's not a whole lot of funding, science research funding is stretched very thin, and it's one of the reasons why academia is a nightmare. Like, as a professor, your existence has to be writing grants over and over and over and over again. And I think the current like rate is like 17% grants get accepted by the NIH. So like your life is just begging people for money. And you know, that's why I'm so grateful to the people who've donated money for my stream or for my Patreon, because I literally use that to fund these experiments rather than write grants when I can. So I still need grants for the more expensive stuff, but for some of the smaller stuff, it's really great.
The more you know.